Okay, so let us start today's class. So in the last class, we have seen about bacterial prokaryotic classification and the Berge's manual of classification. So in that, the first three types we have seen, it is all about the based on the shape. It is all about, we call them as cocci, rod and spiral. These are the most three common shapes, and two are the very most common, that is the cocci and the bacilli, that is rod. So these are the two most prominent shapes of bacteria we can find. And second to that, we have about the based upon the cell wall, how we classify the bacteria as a gram positive and gram negative, and we have just compared the differences and how they can be visualized. So the differential staining technique, that is the gram staining, is a technique used to differentiate the bacterial cells as gram positive and gram negative. So with the differential staining, that is gram stain, we get gram positive bacteria as a crystal, the violet color, as well as the gram negative bacteria as pinkish in color. That is because of safranin dye, and the violet color is because of crystal violet. The purple color is because of crystal violet dye. So this is how we differentiate. So the gram staining is used for visualizing the structure of bacteria as well as to differentiate them as gram positive and gram negative. If you use only a single stain, that is crystal violet, it is called as a simple stain. Either crystal violet or the safranin can be used, and they are called as a simple stain. And we have just seen how this is classified combining both the gram positive and gram negative, and the gram positive cocci, gram positive bacilli, and similarly gram negative cocci and gram negative bacilli. So now coming on to the physiological characteristics of the bacteria, how we can classify them. So first, the category that we are taking is oxygen requirement. We have the numerous physiological requirements for the bacteria to grow, but we will just take out only very few, and one of them is oxygen requirement. So each and every bacteria, they require oxygen, or some may not require oxygen. It is not that all the bacteria they require oxygen for the growth. Okay, so based on this category, we call them as obligate aerobes. If some bacteria, if they can able to grow in, if they require higher amount of oxygen, oxygen is very essential for the bacteria for the growth, and those organisms are called as obligate aerobes. This kind of a bacteria, they will not grow in the absence of oxygen. The figure here is to depict that if you grow the bacteria in a liquid media, okay, bacteria can be grown in both solid as well as the liquid media. So in the solid media, the bacteria will grow onto the top of the solid support. If this is a solid support, so bacteria will be growing on top as a kind of a small button-like stress. Like this, the bacteria will be growing. Whereas in case of when you grow them in a liquid, so the aerobic bacteria, they will float onto the top of the liquid media, because the amount of oxygen availability in this area will be higher. So they will grow only onto this region. So these are called obligate aerobes. And the next category is the one which is obligate anaerobes. That means they are strictly, they do not require oxygen for the growth, and that's why they always settle down to the bottom of the liquid media. If you grow them in a liquid media, they will settle down only in the bottom, because here the conditions will be, there will be no oxygen will be present here. So they prefer to settle down in the bottom of the liquid media when you grow them in a test tube. This is called as obligate anaerobes. So here it requires a maximum amount of oxygen. It doesn't require any amount of oxygen in the media. And here there is another
Okay.
category called, yeah.
So either here growth means growth in size, or by fragmentation it will be divided to more numbers.
It will be dividing in numbers. Growth in of bacteria is majoritarily referring to the increase in number, because their size growth is very minimal. Within 20 minutes, they will attain their full maturity. So every 20 minutes, this E. coli bacteria is a well-known example. They have a 20-minute life cycle, and at every 20 minutes, they will be doubling their number. Okay. So now the facultative anaerobes. So these are a kind of organism which can tolerate to a certain extent, like in the absence of, if the oxygen is limited, still they will be able to grow. Whereas in case of obligate aerobics, it requires oxygen, but here Oxygen is required. If oxygen is there, they will grow very well in this area. If oxygen is scarce, they will just they will grow slowly uh, in the oxygen absent region. But here the growth rate will be slower. Here the growth rate will be higher. But in this case, they will not grow in the absence of oxygen. So this is called facultative anaerobes. And here
So this one
aerotolerant.
once again, with faculty anaerobes. So why some are growing so faster in the upper area and why they are growing slower in the
This is
bottom
why,
area?
because they require oxygen for the growth, but in the
Oh,
absence of oxygen, they will tolerate it. So the growth rate will be reduced.
okay.
So doubts will discuss at the end. So in between, like I think the others will be disturbed. So we'll just at the end, I will dedicate 10 to 15 minutes for discussion. So we'll discuss all the dots over there. Okay. So aerotolerant anaerobes. So these are an a strict anaerobic, sorry, these are anaerobic bacteria which can tolerate a certain level of oxygen. Okay. So these organisms, they will grow, they prefer to grow in the bottom area, and they may also grow in the presence of oxygen. Okay. But they will be preferring to grow in the in anaerobic condition, but they can tolerate the little amount of oxygen. And this is another category called microaerophiles. They require very less amount of oxygen for the growth, so they won't prefer to grow on this top area, but rather they will be settled somewhere below the surface of the media or the growth media. So they will be growing just below the surface of the growth media. So these are called microaerophiles, and they require only very lower amount of oxygen for the growth. So now, based upon the oxygen requirement, these are the 5 different categories we have. Obligate aerobies strictly require oxygen. Obligate anaerobes strictly do not require oxygen. They grow only in the absence of oxygen. Facultative anaerobes, they grow well in the presence of oxygen, but growth rate is reduced in the absence of oxygen. And aerotolerant anaerobes, they are anaerobic bacteria, that is, they do not require oxygen, but they can tolerate a certain level of oxygen. Microaerophiles, they require very little amount of oxygen for the growth. So why this differences they have? Because these bacteria which are anaerobes, they will not have a mechanism to utilize the oxygen. The presence of oxygen could damage their proteins or cell wall. Their enzymes will be they will be damaged if they are exposed to the oxygen. They do not have a mechanism to prevent the oxidation of their enzymes and proteins in the presence of oxygen. Whereas the obligate aerobes and other aerotolerant microorganisms have the capacity to neutralize the oxygen, because oxygen can cause oxidation. So they has a capacity to neutralize the oxygen, and they are able to detoxify it. So they have the ability to grow in the presence of oxygen. So this is a first physiological classification based upon the oxygen requirement. And now, based upon the nutrition, how bacteria can able to synthesize its own food. So based on this, we have a different classes. So if you look at the nutrition, there are three important essential nutritions are there for the bacteria. First one is the carbon source. So what is the source for the carbon of the bacteria? If the bacteria can able to utilize the CO2 present in the atmosphere, they are called as autotrophs. Okay, because they do not rely on other organism for the carbon source. So if they use atmospheric CO2, they are called autotrophs, as like in the plants. And in case of heterotrophs, they rely on the other organic they rely on the organic carbon instead of CO2. So organic carbon, where it is available, it is available in the autotrophs, because autotrophs, they fix the atmospheric CO2 and they convert them into an organic carbon. So heterotrophs, they rely on the autotrophs for the carbon. Okay, so these are organic carbons that are fixed from atmospheric CO2. And next, the requirement is the energy source. What is the energy? That is, what kind of energy they utilize. So one group of organism which uses light, they are called as phototrophs. The light, the term photo is used to mention the light. So because they utilize the light, they are called as phototrophs. And the second group of organism called chemotrophs, because they use the chemical energy. So they oxidize organic and inorganic compounds that are available in the other organisms. So by doing such kind of an oxidation, they are able to generate the energy that is required for reducing or metabolizing or producing utilizing this carbon sources. Okay, so that's why they're called chemotrophs, because they use the chemical energy that are derived from the organic or oxidation of organic as well as inorganic the materials that they have. And next, the electron sources, based upon what is the electron donor they have. Okay, during the process of oxidation they require the electron donors and acceptors. So based on this we call them as lithotrophs, which uses inorganic molecules, and organotrophs. The organo means organic. Okay, so this is, or organotrophs, they uses the organic molecules as an electron acceptor. So this is the three broad nutrition or the energy source, carbon source and electron source required for their metabolism. So based on combining all these three, we were able to classify the organism into multiple groups. So as a overall, like we will classify them as that same table is represented over here in this flow chart. We have, based upon the sunlight and not sunlight, phototrophs and the chemotrophs. So chemotrophs are always the heterotrophs. Okay, so I will just discuss in the next slide in detail like how we can combine them. So if you look at here, the nutritional types are photolithoautotrophs. The photo means they use light, litho means inorganic electron donors are used, and autotrophs, they utilize the carbon source, that is the atmospheric CO2. So based on the name, we can able to guess what kind of a nutrition the organism would be requiring. So in case of photoorganoheterotrophs, they use organic molecules as an electron donor as well as acceptors. Heterotroph, they rely on the organic carbons from the other organisms. And phototroph, photo, that means they use the light energy for their oxygen reactions. They use a light energy. So examples are given here. You can just remember any one name for each of this category, though there are given many. These are not the bacteria name, actually some group of bacteria. Okay, this purple and green sulfur bacteria, they can able to do photosynthesis. Cyanobacteria, this purple and green sulfur bacteria, they contain the pigment in them. The pigment are the light harvesting complexes they have. So those pigments help the bacteria to trap the light energy for utilizing the atmospheric carbon, and they can synthesize their own food that they're required for their own metabolism. So here the photoorganoheterotrophs, we have the purple non-sulfur bacteria or green non-sulfur bacteria. Though they have the pigments to utilize the light energy, but the carbon sources, they rely on autotrophs. The organism which can fix the atmospheric CO2, they rely on them for the carbon, and they utilize those organism as a organic carbon for their growth and metabolism. The chemolithoautotrophs, again, chemo means they use the chemical energy for the energy as well as the electron source. Litho is inorganic electron donors or acceptors are used, and autotrophs, they can able to utilize the atmospheric CO2. So this is how we have classified the organism like chemolithoheterotroph and chemoorganoheterotrophs. So these are the 5 major nutritional types we have, and based, if you understand this 3 categories very well, and we can able to explain these categories with the help of the name. Okay, so now moving on to the next slide. So this is all about the physiological based classifications, and now one of the modern system of classification. So here we use a specific molecule called 16S rRNA. Okay, the 16S rRNA we will see when we study about when we read about the ribosomes. So when we go in detail about the ultrastructure of bacteria as well as any eukaryotic cells, we will have the ribosomes. Ribosomes contain this 16S rRNA. Okay, so 16S rRNA is a kind of a nucleic acid. By determining the sequence, what kind of nucleotides they have, ATGC, like that, they will have a certain number of nucleotides. By determining the number of the nucleotides in this RNA, they have been able to classify the organism to 3 broad domain. So earlier we have seen the 5 kingdom classification, where based upon different kinds of properties, the organism has been placed under 5 different kingdom. But here, if you look into the 16S rRNA sequences, we can only able to define 3 kingdoms. Those 3 kingdoms are eukarya, which includes all animal, plants and fungi, all comes under this eukarya category. And next, the prokaryotes. So we can simply say eukaryotes and prokaryotes. Okay, in the prokaryotes, it can be further able to distinguished into bacteria and archaebacteria. Archaea. So archaea we will see now. Remember this term, hyperthermophiles, methanogens and extreme halophiles. So these all we will see in detail. Okay, so this kind of a classification was introduced by the WHO's in the year 1977. So this is based upon this 16S rRNA sequences. So you want to remember only that based upon this three domain system, we have prokaryotes and eukaryotes, where the prokaryotes are further divided into bacteria, archaea, and the eukaryotes as eukarya. So these are the three domains we have based upon this 16S rRNA system of classification. And now coming on to archaea. So earlier we have seen the prokaryotes in general, and whatever we have studied the classifications is all about the common this bacteria. Only this bacteria we have seen the classification of this bacteria. Now we will see into this archaea, that is archaebacteria. So this archaebacteria are the domain archaea is entirely distinct from that of the bacteria. They have many dissimilarities with the bacteria, though they are like have most similarities, but still some of the features makes them very distinct from that of bacteria. So the very prominent differences is at cell wall. So earlier we have seen that cell walls in bacteria, they contain peptidoglycan. So here in case of archaea, so no such peptidoglycans are present in the cell wall, and they have ether-linked membrane lipids. There we have the phospholipids, and here we have the ether-linked membrane lipids. So for the sake of understanding, I'm just showing these examples. You just want to be sure that they have difference in the cell wall. Okay, there is a major difference between the cell wall in the archaea and the bacteria, and also they have a different physiological features also. For example, in this archaea, they further have different classes, some called as like Euryarchaeota, Cranarchaeota and Coraarchaeota. So these are the three major subdivisions of archaea. Okay, and in these subdivisions we have further the bacteria subdivided into methanogens and extreme halophiles. But you don't need to remember these names. Euarchaeota, Cranarchaeota, and this kind of three names are not required. But I would require you to remember this methanogens, extreme halophiles, and hyperthermophilic sulfur-dependent prokaryotes. So these three terms are very important. So these three you have to remember. So further, this archaea has been classified into this Euarchaeota, and the Coraarchaeota. And among this, we have the subdivisions like methanogens, extreme halophile, and the hyperthermophiles. So we'll see in detail what are they. So this is what the subdivisions already have shown you, this U, RQ, and the Granarchata, so and all this. Okay. So now coming on to the physiological classification of archaea. Okay, so based upon the physiology, they have been classified already. I told methanogens, extreme halophile, and extreme thermophiles. So the methanogens are the organism that produce methane. So these organisms, they do not require oxygen for the growth. Okay, they are able to grow. They are a strict anaerobic organism. So we have seen already in the bacteria, we have the classes as like aerobic, strict aerobe, facultative anaerobe. So the likewise we have the 5 different categories. So this methanogen is strict anaerobic microorganism. They grow only in the absence of oxygen, and they can able to produce methane. Okay. And these organisms are the one which produces the biogas in our the biogas plants, and this organism produces the methane which is used as a fuel. And next, extreme halophiles. These are the organisms which grows in the very high concentration of salt. So we know that in general we use salt as a preservative in preserving many items in our household, because salt, excess amount of salt can inhibit the growth of bacteria. But this extreme halophiles are a group of bacteria which will grow only in the presence of high amount of salt. Okay, and extreme thermophiles, so these are the organism which grows at a very high temperature. In general, we know that when you boil a water, we can able to kill the bacteria, but we cannot kill this extreme thermophilic bacteria by boiling the because they only grow in this high temperature. So they need very high temperature for their optimal growth. At very low temperature, these bacteria will not grow. So we'll see in detail where we can see these this kind of extreme bacteria. So methanogens. So as I said already, these are the organisms that are responsible for production of methane. So in general, the cow that is being bred also contain this methanogens in their intestine. So they have a intestine which is being populated by this methanogenic bacteria, which utilizes whatever the cow feeds on. So those the carbon sources are being utilized by this methanogenic bacteria, and they can able to produce methane. Okay, and the cow, when they release the methane gas. Okay, so if you take a simple comparison, a two cow can release about 504 pounds of methane, which is equivalent to the carbon dioxide which are produced by a single the petrol or diesel driven vehicle. So methane is also, we know that, a greenhouse gas, and the carbon dioxide too, but methane is highly, like it is more it can cause a lot of effect than the carbon dioxide. So as I said already, they are obligate anaerobic organism. They will not tolerate even a brief exposure to oxygen. So in the cow's intestine, they do not have the oxygen. Even in humans intestine, we have some sort of, we do not have the methanogens, but we have the bacteria that are anaerobic in nature. They are normal bacteria, they're not a archaebacteria. So those bacteria survive in our intestine, that is providing them the anaerobic condition. So if you look at the anaerobic conditions, wherever we can see, it includes the marine. In the deep marine area, there will not be available of oxygen, so they can able to grow there. And freshwater sediments. So we know that the marshland are the example for this growth of, like the oxygen devoid environment in the conditions. So in the marshland, in the deep soils or the muds of a marshland, these methanogenic bacteria can able to grow. The examples are that a rice field. The rice field is flooded with water, and in that condition where there is no oxygen, these methanogenic bacteria can able to grow, and that is why the cultivation of rice releases methane into the atmosphere, because these organisms, they release methane. And deep soils, and again the intestinal tracts of animals I told already, and the sewage treatment plant, because sewage contain huge amount of organic matters are present, and they become the sewage containing facility or the sewage ditch they contain, they will not allow the oxygen to get into the deep areas. So the methanogenic bacteria can grow there, and we would have heard like the methane poisoning is being quite common when the sewage is being cleaned up. So this all because this methanogenic bacteria, they grow up in the sewage, and they can produce, and they were able to produce the methane. And we would have been noticed in the sewages there will be a small bubbling up here. Those all because these bacteria, they metabolize and they release the methane. Okay, and metabolism, how these bacteria they do metabolize, they use hydrogen as an energy source, and CO2, whichever is available in the growing media, they use the atmospheric CO2 as a carbon source. They are autotrophs, and they use hydrogen as an energy source. So in the process of metabolizing this hydrogen and CO2, they produce methane, which is released as a gas from the growth environment. Okay, so, and the methanogens are normal inhabitants of animals. Like the animals, they able to metabolize this cellulose with the help of this methanogens only. And the cow releases about 50 liter of methane a day during the process of when they're chewing the cud, like when they do digest, when they do second time, they used to chew the grasses or whatever the fodder they feed on. That is called chewing the cud. Erectation is called the second time they take out the grasses from the stomach, and they will be chewing it up. So they release methane through their mouth as a belches. Okay, and methane is a significant greenhouse gas. So these methanogens, they contribute to the release of methane gas in the atmosphere. So these are the places where we can see the rice paddy field. I said it is flooded with water, and it forms a perfect anaerobic condition for the growth of this methanogenic bacteria. And they, by decomposing the matters, like the organic matters, whatever they are in the below the water level, they produce methane. And rice fields are example, like they can contribute to the greenhouse gas rise in the atmosphere. And here it is a sewage area where we can see the small bubbles that are coming out of the sewage. These are methane gas pockets that are released by the methanogenic bacteria. And the biogas plant is a very perfect example for the methanogenic bacterial growth, because here they utilize the carbon, the CO2, whatever present in there, and they are converted into methane. So this is just, you can ignore this. This is how the methane is produced by means of utilizing the CO2 and the energy sources. So we can just leave this, only it's an examples. And the second category of archaea is the extreme halophiles. So the halophiles means they live on high salt concentration, that is NaCl. So they can grow as high as 25% NaCl. If a water contains 25% NaCl, even in that condition, they will be able to grow. And they cannot grow in the very low level of salt condition, that is as like our normal tap water as well as the groundwater. These organisms, they cannot grow. They always require a high amount of sodium chloride for the growth. And these organisms can be seen growing in the Dead Sea, the Great Salt Lake, and or the evaporating ponds of seawater, where the salt concentration would be close to this 25%. So in India, we have the Run of Kutch, where we can find the salt, the white salt, the patches will be there. So we can able to isolate this extreme halophiles in that environment. We would be able to do. And these organisms, they use light energy for the production of, for their metabolism. So they use a pigment called bacterial rhodopsin. So this is a big pigment. The bacterial rhodopsin is used for the energy production. So with the help of this bacterial rhodopsin, they can able to capture the light energy, and they able to pump the hydrogen ions across the cell membrane for the production of ATP. So the imbalance in the hydrogen ions across the membranes of the bacteria, it produces a gradient which helps in the synthesis of ATP. You can just remember the bacterial rhodopsin is helpful in synthesizing the ATP by harvesting the light. So this is the function of this bacterial rhodopsin. Whereas in plants, this is done by chlorophyll pigment, and they use water for the hydrolysis of water is done, and oxygen is evolved. So here there is no oxygen has been evolved. The example of the bacterium is Halobacterium halobium. This is an example, and they adapt to the high salt environment, and they have a purple membrane. And because of this pigment, bacterial rhodopsin only, in the salt land, in the salt lakes, saltons, we can able to see the different colors. The colors are basically because of these bacteria which are growing there, and they have this color of purple yellowish pigment. So that able to be seen as a purple saltons in the area, these areas. Okay, so this is all about this extreme halophiles. And next, the thermophiles. So the thermophiles, the thermo, it is at temperature. So they grow at very high temperature, that is close to 100 to 80 to 105 degrees Celsius for the growth. So they require this temperature for the growth. At normal room temperature, this bacteria cannot grow. So these bacteria can be seen only in the hydrothermal vents as well as in the hot springs. So in those area, the temperature is always maintained close to boiling temperature of water, that is above 70 or 80, 100, up to 100 degrees Celsius. And these organisms have evolved in such a way that they can able to withstand such high temperature. Their enzymes and the membrane structure, or else other proteins, whatever they have been evolved in a way to withstand such high temperature conditions. Okay, so the example or this thermophiles are the sulfolobules and the thermus aquaticus. These are the two organisms that are very well known example for this hyperthermophilic microorganisms. And the environmental conditions where we can find them is sulfur rich environments, that is usually associated with the volcanoes and hot springs. These all associated with volcano only, geysers and the fumaroles, and the deep sea where we can find the smoky vents. Okay, These are the area where we can able to see these organisms, because the temperature close to this environment will be close to 80 or 100 degrees Celsius. So examples have said, these two are the examples: sulfur and the thermus aquaticus is the organism. And here the optimal temperature is an example. We say it as 70 degree, whereas for the normal bacteria that we routinely see, they have optimal temperature of close to 27 to 30 degrees Celsius. Okay, and based upon the temperature also, we classify the organism into different categories like mesophiles, thermophiles, and there are so different. These are one of the physiological factor. Okay, but we won't see in detail. So these sulfur they grow at as high as close to 90 degrees Celsius in the sulfur-rich hot acid springs. So this is all about the thermophilic microorganism. And so now it is all about like the ultrastructure of prokaryotes. I will introduce this slide and I will wind up here. And tomorrow see about each and every the structure of a bacteria we will see. So earlier you have seen about the shape of the bacteria, size we have seen, and the nutrition also we have covered. Like only very few factors related to the nutrition we have like the types of nutrition we have discussed. The physiological factor I have discussed only about the requirement of aeration, based on which how the organisms are classified. But there are so many other factors, like based upon the pH requirement, the organism can be classified. Based upon the temperature at which they grow, they can be classified. So there are numerous other ways the physiological conditions can be used to classify the organism. And now we will see about some of the very fine structure of the bacteria, like the cell wall, plasma membrane, how do they, what are the different kinds of organelles they have. So all those things we will see in detail in this session. So these are different types of bacteria. You just ignore this, no need to remember in any of this, but just I want to introduce, I can just say like eubacteria are the true bacteria that we have, like E. coli, bacilli, whatever the bacteria that we are talking about till now, they are all called eubacteria except the archaebacteria. They're called eubacteria. And actinomycetes are a kind of a bacteria, but it is not a true bacteria. They have a different morphology when compared to the bacteria. So these actinomycetes are responsible for that smell of soil, where whenever it rains, we can able to smell the soil. So this is because of this actinomycetes. Upon contact with the soil, they produce some kind of a scent which we feel as a smell of soil. Mycoplasma or a very small bacteria kind of a thing. Myxobacteria is a very rare kind of a bacteria which we don't see routinely. And chlamydia, rickettsia, and cyanobacteria, these are some specialized group of bacteria. Okay, but mostly whatever we have seen is all about the eubacteria apart from the archaea. So this is an ultrastructure. With this we will wind up. So we know that if you go from the outer side, the outermost layer in a bacteria is the capsule. This is the outermost layer. Capsule is not seen in all the bacteria. Few bacteria they have capsule, and few bacteria they may not have capsule. So capsule is not a very essential factor for the bacteria, but the presence of capsule adds some advantage to the bacterial properties. Okay, so the capsule forms the outermost layer of the bacteria. And on the capsule we have a structures called fimbriae. These are like finger like projections, as you can see, a finger like projections out of the cell wall. These are called fimbriae, and this has multiple functions. They use this fimbriae for movement of the bacteria, as well as this fimbriae can be otherwise called as a sex pili it is called as, because they use this kind of a structure to communicate between the two bacteria. Two bacteria, they can share their genetic material with the help of the sex papilli, which used to establish a connection between them. So they transfer their DNA between the bacteria via this structure. Okay, they can able to transfer. And up next to the capsule we have the cell wall. Okay, so the next to the capsule, this layer is cell wall layer. Cell wall, again we have seen the two different types of cell wall, the gram positive, gram negative, that we will see in detail. And next to the cell wall we have plasma membrane, that is the innermost layer of the outer covering, that is cell envelope. All these three This capsule, cell wall and plasma membrane can be called as cell envelope. Okay, and this envelope encompasses this area which is called as cytoplasm. This area is entirely called as cytoplasm, where we find nucleoid, the DNA containing part of the bacteria. It is called as nucleoid. Otherwise, we have this ribosomes in the cytoplasm, and inclusion bodies. These inclusion bodies are a kind of small granule-like structure. Excess of protein or lipids they will store in the form of this inclusion bodies. Okay, so these are some of the very essential and important organelles, or we can see in the bacteria. And one more organelle is the flagella, which is a kind of tail the bacteria they possess. They use it for movement. Basically, the bacteria which has the flagella can able to move from one place to another place in liquid, as well as in the in solid environment. The fimbria helps them to glide over the surfaces. Okay, and flagella is not a compulsory requirement for all the bacteria. As I said, similar to the capsule, flagella is also optional. Some may have flagella and some may not have flagella. Okay, so this is all about the ultrastructure. We will read about each of the structure in detail in the upcoming classes. So now we can have a discussion on whatever today I have covered.
Sir,
Yeah, just a minute. I will just stop the recording and we'll discuss.